Hello everyone, thank you for joining us today. Our presentation is on population estimation techniques of the bobcat, Lynx rufus. So the problem is that mark recapture experiments rely on capturing marked individuals during subsequent capture sessions. However, with secretive mammalian carnivores such as bobcats, recaptures can be difficult, costly, and time-intensive. Previously, harvest records have also been used to estimate bobcat populations. However, this method is not always effective and cannot be used in all studies. This has led to inaccurate population estimations in most states within the United States. We analyzed 3 common mark recapture techniques to determine which method will provide the most accurate population estimation while also being the most efficient and cost-effective. Our first method is trip and infrared cameras. This method consists of using an array of remote-triggered cameras. And with these cameras, researchers non-invasively capture the study species through photographs, and then they use these photographs to identify individuals based on their natural markings. Encounter histories for marked individuals can then be constructed for any recaptures obtained from camera traps, which can then provide a detection probability and population estimation. Their pros are that they're low cost, low maintenance, and are non-invasive, and the photos can identify individuals through natural markings. As well, they require limited field effort. These are camera trap photos of the same bobcat. You can tell this by the marks on its tail and its leg, which I have circled. There are many other identifying natural markings as well. However, the cons for these cameras are that they need a high density of cameras in order to capture enough individuals to obtain the data needed. The graph on the right demonstrates this quite well, with more photos being captured as the camera density increases. In addition, adult offspring ratios may be biased due to decreased mobility and visibility in offspring, and camera view can be restricted by thick vegetation or heavy rain. And I want to expand upon this last con for a moment. So this is a photo from a camera trap, and you can obviously see that there's some kind of animal behind this tree and these branches, but it's extremely difficult to make out. This can make the data collected by this camera unusable. Our second method is fecal DNA, and this method utilizes scat of the study species, which is collected and then genotyped to identify individuals. You can also identify territory and home range use. With this information, you are able to estimate the population size using mark recapture techniques. Collections can be done in multiple ways. However, the 2 most popular are humans using line transects or with the use of dogs using a grid cell system. The pros are that fecal DNA allows you to identify individual genotypes, it can identify territory, it's non-invasive, and it does not require the use of attractants, which can potentially minimize bias. The picture on the right is an example of scat transects used in a study on marmots.
Some of the cons for fecal DNA: analyzing scat data is more costly than other methods like camera detection. Human and dog detection is not 100% foolproof, and often there are non-targeted scat collected, and also, as you can see from the picture, a dog found a turtle. Rain and moisture can have negative impacts on detection for dogs, so after it rains it kind of loses its scent and the dogs really aren't able to smell the scat as well. So for our next method, we have the hair snare method. Hair snares can be set along trails, they can be set on trees. It's essentially anything that can take hair off the animal that can be collected for further DNA. Hair snares are often able to determine more than just DNA. Extremely cost effective to use. Researchers can leave their hair snares out for long periods of time in the field without checking them, and there is also very minimal training required to use hair snares. So for some of the cons to using hair snares require the use of attractants, which can potentially bias the study. Bobcats often do not exhibit rubbing behavior as often as other animals would. Cross-contamination of samples is prevalent, as many individuals can use them before they are checked. So you might have a lot of different hair follicles in one. Single hairs may yield insufficient data for DNA, leading to high genotyping error, and they require a high density of individuals in the area to obtain the data needed for the study.
For recommendations, we recommend the use of trip and infrared cameras. Based on our sources and our meetings, this was overall the most accurate, cost-effective, and efficient method compared to all the other methods.
Also, with advances in technology, cameras are constantly getting better. We're getting better photos, which leads to better data. So only time will tell how much better this method will become in the future. And I just want to quickly thank all the sources that we used for our presentation, as well as our judges and all of you for watching our video. If you have any questions or comments, go ahead and comment down below. And thank you once again.